Done. It is showing here.
You should hit, yeah.
Oh, it's showing. Where? Am I audible? No.
No.
Am I audible? Zoom here. Zoom. Come here.
I see that.
Are you live?
We go to sport. Yeah.
You go down.
Yes. Okay, fine. Yes. No sound.
Okay.
शुरू नहीं किया शुरू नहीं किया ठीक है अभी ठीक है शुरू कर रहा हूँ ठीक है कैमरा पे तुम्हारा
Sौम्य.
सौम्य शुरू कर रहा हूँ ठीक है सौम्य ओके so good evening everyone welcome to today's talk on prebiotic chemistry on behalf of the NISER-Astronomical. I am Adesh Natthuji Thawale, 5th year integrated master student from the School of Chemical Sciences, and I would like to thank all of you for coming here and attending this talk. This is quite a big thing for me that you all came, and I see people across disciplines come here and attend my talk. There are people from mathematics, biology, even CS, and that is a big thing for me. So, prebiotic chemistry, I would give a little disclaimer beforehand that prebiotic chemistry is an interdisciplinary science, which means that it involves practices from biology, chemistry, and all other sciences. So, if you are an expert in any of those, but I would really love to discuss what now, but not right now. Okay. If I make a absolutely wrong statement, you are free to raise your hand now. But if it's not something major, you can just okay. I do not claim expertise over any of those fields that I just mentioned. Okay. Uh, right. So we'll begin now. And for all those, all of you ask your questions at the end, so that we have the presentation. Okay, we need to feedback. Okay. Everything. Is it okay now? Audio ठीक है?
ठीक है.
मैं जोर से बोल दूंगा. ठीक है. Okay. So we'll start right away. If you have no questions for me right now, you have small questions you can ask me in the middle of the presentation. If there are major questions, keep them handy. Okay. So I'm sorry, I have 100% and that from my. Okay. Anyways, life, as we know, is very complex, and we hear it a lot at NISER. Your life is absolutely complex. And as simple as it's okay to be complex, right? But when you keep on saying the same stuff every day, that life is complex, we start to question what is life. And I would like to ask this question to all of you. What is the definition of life? And I would really appreciate some answers from our audience. How would you define life?
Thing that can replicate and pass on to next generation.
Okay, fine. Any more answers? Metabolic something? Divya, you had something?
The same thing, metabolism.
Okay. Any more answers?
Something that's not dead.
Something that's not dead. That's nice. It's a circular argument, but fine. Dead is not dead is technically correct, but no, not really. Anyways, Ava,
Yeah, the same thing.
that is not dead.
No, the one that reproducing.
Okay, reproducing. As in, life recreates itself.
It's one defining factor of life.
Okay. And you're not wrong telling that. All of you mentioned some characteristics of life. If we go to the dictionary, life is defined as a condition that distinguishes animals and plants from inorganic matter. And then they mention some of the characteristics of life. Okay. It includes the capacity for growth, preceding death. Okay, so all of you were writing some domains. But there's a catch to this definition. Made when we already have a lot of things alive right in front of us. Okay. What I mean by saying this statement is that this is just a definition that we have made up based on certain characteristics that we saw around ourselves, and then we classified all of the things that had those characteristics as being alive. So we had examples to define life. But what if we didn't have examples to do that? How would you define life then? If you really don't have examples of it, how would you define life? That is a very important question in the context of the study of origin of life. What about a prebiotic context? Imagine a world where there is no life, not negative. If you find something on that world, how would you define whether it's alive or dead? Right? You just listed out the characteristics that you see see around yourself. Reduce definition. For all you know, the other planet might have some other characteristics which which objects on that planet show and might be common to some objects on that planet. So you cannot really say that life has a fixed definition. This question can also be asked in a similar way. When does a thing transition from being dead to being alive? Think about the origin of life. We are not concerned with how it began. Just think about the origin of life. If you have something, if you have some object, when does it start becoming alive? What are the characteristics that it must show? We listed those in the last one, but we really don't know at this point of time whether it's really alive or not. It's just something that is performing some sort of complex reactions. We don't know whether this will create life or not. So, this tells us that we really don't have a fixed definition of life in the context of its origin. So, how do we study something that we really well? How do you study something that you cannot define precisely? I hope people from CS and physics and mathematics can answer this. You do not have a fixed definition of something. How do you study it?
I would say you don't answer.
I am asking mathematicians and physicists.
So we don't bother.
This is what we practically do. We assume something, right? We assume something. There's one more thing to this. How do we study something that we cannot define precisely?
We define something and then we
Right? Comparison, approximations. Right? We make assumptions and we make approximations. So, CS has a course, approximation methods, I guess. In that course. Anyway, the point of science is to approximate and make assumptions. Because nothing is defined in a very precise way. Very few things are. So, the answer is assumptions and approximations. And in this context of study of origin of life, we have to make certain assumptions and certain approximations. So, the first assumption is that evolution. So, evolution is really fundamental to all of the study that we do regarding the study of origin of life. Evolution, as we know today, is an exact science. It's close and it's a fairly valid assumption, I think. Is it a valid assumption? People who have studied evolutionary biology, is it a valid assumption? Biologists, there are so many biologists. Is this true? Can you take this assumption to be true?
assume they said something.
Yeah, I'm going to start picking names now. Is this a valid assumption to do? Okay. And for all of those who do not haven't taken the course evolutionary biology, please take not right now a very good course, but use okay. It can only build upon existing organs. That is the summary of evolution. And then exact aim of evolution, as in what it works towards, is the survival of the species. It does not ensure the survival of an organism individually. If there is a cluster of organisms, if there is a colony, evolution will try to ensure that the colony survives. It will not try to ensure that one single organism will survive. This is also very important. And first thing that I said is that it cannot add new organs. It can only build upon those which are already present. I hope this is correct, biologically speaking. So this is the assumption that we take. We still are missing approximations. And luckily, we do not need to look far for example. So how do we take approximations? We look around ourselves, see where this thing is being applied, and then we try to approximate it, right? Okay, so we are very fortunate that we do not have to look far, or we do not need to look very extensively, right? Here is Pulkit with my batchmate. He will also be the representative of all life that is present on Earth right now. Also the life of NISER, he is also the life of our batch. He is also a new boy with that element. All life present on Earth. Okay. When I mean Pulkit, I mean all life present on Earth. Okay. And for some regard, Pulkit came from a single cell. Now this here is an eukaryotic. Okay. This example here that I have given is an eukaryotic cell. Okay. Pulkit came from a cell. That is very evident. Right? All the life that you see around us came from a single cellular organism. That is something that you have learned fairly commonly in textbooks. Biology textbooks always make sure to remind you that you came from a single cell. Right? Not we had a question that why is the arrow opposite? And if you have taken C102, you might know the answer. It's a retrosynthetic arrow. Right? What does retrosynthesis mean? It's an arrow that depicts things that precedes them. Okay, so the direction of the. Okay, so if I have a molecule C which is being made C to A. Okay, that is the retrosynthetic arrow. Pulkit came from a single cell. Right? Just because Pulkit, the person, is an animal, as a but and we have evidence for this that life, how life evolved. Right? So if you, it's not very clear here, not get a better image, but you can see that eukaryotes evolved around 2,000 okay, on the earth. Right? So here is the evolution of eukaryotes. But then eukaryotes are not the only animals, only organisms present on Earth right now. There are other organisms as well, and probably shed some more light on this. What other organisms are present on Earth right now, apart from eukaryotes, on the same level of classification?
Prokaryotes, archaea.
Okay, we have answers: prokaryotes and bacteria, right? So all of them evolved at some time. We saw where eukaryotes evolved. All of them did evolve at some time. And there's something that connects all of them. There's something that connects all the species present on Earth today. I'm not just talking about eukaryotes. What is that? Biologists and leaders again. This part will be very biology focused. The next part will not that much biology focused. So I'm really sorry about that. Yes. What is the thing that connects every organism on Earth? Evolutionary. DNA, RNA is fine. That's not the answer I'm looking. Evolutionarily speaking, what is something that connects every organism present on Earth? I had one answer. Gupta. That is the correct answer. Last universal common ancestor. Okay. Now, if you have heard this name, you might have also seen something like this. Right? Luca is the origin of all extant life on Earth. I can make an attempt to explain, but the time. Just know that Luca is from where all of the organisms that you see came. And this is how they represent it. Luca is the root, all the other things are branches. Okay. So this is not a complete why is that? That is because something like this happens. Okay. So Luca, you can say that for simplicity's sake, we had something called Luca, but it need not be that simple. Something like this happens all the time in evolution. Like if you have read a little bit about the cell, you might know that mitochondria and the cell have different sets of DNA. So mitochondria has its own set of DNA, and cell has its own set of DNA. Okay, and that was due to the mitochondria being another organism which came over to a eukaryotic cell and then got assimilated. So this DNA that the mitochondria has is proof that it began as something else. Okay. Yeah, so the actual tree would look something like this, but this is also incomplete. Why is it incomplete? Because first of all, we have no information of what lies here. Okay, at the base of everything. We have no information of that. This tree gives us nothing. A more complete picture will be given by something like this. Okay. And because you see, Luca is very far off from the origin of life. Okay. Here you can see that all the extant life on Earth is highlighted. So you can see there's one branch that evolved into eukaryotes. Okay, one branch that evolved into archaea. But there's a single stem that connects all of them. Luca. Okay. We just saw in the previous image that it's not as simple as this, but this is a very zoomed out image. This is a very zoomed out tree. If you zoom out large enough, you'll see something like this. Now we know a lot about Luca. And why is that? Why do we know a lot about Luca? Think about the assumption that we made.
It's a common ancestor.
It's a common ancestor, fine. It's in the name, it's not a good name. But we know a lot about Luca and very little about the origin of life. Think about the approximation and the assumptions. You said something, I couldn't hear it.
We have some form of life from which the other species have evolved. That's why.
Okay, so we made an assumption that evolution works, right? So if we evolve from Luca, we can just look at all the life that is present on Earth. We can see what characteristics they share, and then we can make a fairly good assumption that okay, since this characteristic is present in all the life that we see on Earth, Luca must have had it, because evolution cannot build new characteristics. It can only improve upon existing ones. Right? So if Pulkit does something, and Pulkit is a all species on Earth, we can be pretty sure that Luca did it as well. Okay. And this is what we'll be exploiting. Okay. This tree also tells us something, that Luca is very far away from the origin of life. Whatever Pulkit does is very complex. Whatever Luca did was also very complex, because all the characteristics that Pulkit shows, they are fairly complex enough to say that okay, when Luca did those, at that point also those were very complex. But we have a hint. The hint is that evolution works. So if Luca did something, we can be fairly sure that okay, at the origin of life, whatever might have happened had the same set of characteristics as Luca has, and by extension we have. Okay, so the study of origin of life is not just the study of how things started. It's the study of what characteristics are essential for us to be present here. Primitive fashion. That is the study of origin of life. Study the characteristics that make us alive. Find that we are alive. And these characteristics in a primitive environment, if you study those, that's what the origin of life studies deal with. Okay. So, according to the assumptions, evolution works. And Pulkit has certain characteristics. So we'll take a look at some of those characteristics and we'll see what kind of studies are done in those. I'll not give a very detailed picture of all of these studies. I'll give a very small flavor of those, but you are ready. If you want, you can explore more. You can ask me. I can provide more references. It will be a little time consuming to go through all of that. This is a study of that. You see, Pulkit was created from something like this, from mess like this. Okay. This here is the chemistry lab, and Pulkit was, if you assume that the chemical origin of life is correct, very messy workstation, that is our Earth, and we have Pulkit now among us. Okay. It's also called abiogenesis. It's an old term, but yeah, it's not used anymore because the other one is more cooler. Degree says cool. Okay. So when I said that Pulkit has certain characteristics that we will study, some very basic characteristics, we'll look at those now. Okay. These are the different types of studies that are done under the umbrella of origin of life studies. Okay. The first one is fairly simple, Miller-Urey experiments. And there's no Pulkit here. Thank you. But the other three are characteristics of Pulkit. Okay. And we'll look at them in a moment. First, we'll go to Miller-Urey experiment. Can anyone here tell me what the Miller-Urey experiments were? You might have learned this in Bio 102 or Bio 101, I don't remember which, but you have definitely heard of it. So I would like someone from the audience to tell me what it is. What is this experiment, Miller-Urey experiment?
Last year we performed an experiment in heat and in old Earth system, how it was. So we performed it in that, and from some bio amino.
Got it right. Miller and Urey were two scientists who did a very special experiment in 1950s that started the entire study of prebiotic chemistry. Okay. They were the original proponents of the chemical origin of life theory. Okay. And this is important. Like we described, they get early Earth conditions in the lab. Different simple chemicals reacted to form more complex chemicals and see whether these chemicals that were formed have relevance to biology today. Okay. So what are the most relevant biomolecules that we know? Simple ones, not the more complex ones like DNA or DNA, not those. Okay. Amino acids, protein, carbohydrate, a little too complex, but carbohydrates is fine. Sugars. Nucleic acids, that is also true. We have amino acids, we have sugars. Nucleic acids, what else? Fatty acids, that is also true. That is excellent actually. Because we do not tend to think about fatty acids. But anyway, fine. So all of these are bio molecules, right? Simple ones, not the complex ones. These are essential to life, as we know it right now. What Miller-Urey wanted to see, Miller and Urey wanted to see whether any of those molecules would be formed in an early Earth system. Okay, and this is what they did to simulate early Earth conditions. Okay, so here you can see Stanley Miller and Harold Urey. I hope it's visible. This is their setup, and you might have seen this already. If you haven't, I'll walk you through it. This bottle, there's a collection of gases in one of the chambers. It is subjected to like electric discharge. It is subjected to heating and condensing again and again to simulate rainfall. And early Earth conditions, like whatever the gases were present on early Earth during the time of evolution of life, those are present in this set. Now, an important thing to note here is that atmospheric scientists have led us to realize that this is not exactly the condition that was present. Okay. They made a mistake in choosing the gases, which has been rectified. but regardless of that, we will stick with the original mirror unit experiment. Okay. This is the setup. They ran it for, I guess, 2 or 3 months, if I'm not wrong. And they analyzed the products that were formed from this setup, this experiment. And to their own, you can see some of the reactions happening here. Observe. Okay. Even here, in the bottom of the screen. However, there's something very unique about that experiment. And what is that uniqueness? So, in 1970s, there was a meteorite that came to Earth, landed in Australia. It's called the Murchison meteorite. Landed in Australia, and scientists were able to take samples of that rock and see what are chemicals that fragment contained. They were looking for biosignatures. What are biosignatures? Biomolecules, simple biomolecules. And to their to their amazement, they found out that there is a correlation between the amount of some of the amino acids present in the Miller-Urey experiment and correlation, but it's still a correlation. Remember that. One more thing, when you are studying prebiotic chemistry, we are not concerned with the quantitative yields of each step. We are more concerned with the qualitative yield. Whether this reaction is happening or not, that is our ultimate question. That's it. Anyways, you can see that some of these molecules have a very good strong correlation with the correlation. Okay. So, study of such experiments, the Miller-Urey type studies, are done to the early Earth atmosphere that was present when life on Earth. They try to simulate those conditions subjected to various processes, lightning strikes, volcanoes, heating, cooling, everything that you can think of. And then they try to characterize the results that they get. All the chemicals that they obtain, they try to characterize it and see whether which experiment leads to better biosignatures. Okay, biosignatures, biomolecules. Right? So this is the Miller-Urey type experiment. Okay.
What do the dots represent?
Dots is how abundant they were. Like the abundance of that species in a given sample. Like 10 ml, that how much? Concentration basis. Okay. So this is fairly obsolete now because this is like turning the light off and throwing darts at a dartboard. Because you really do not know what you'll end up with, and it's very hard to quantify the results of such type of experiments because it's a very complex mixture. And the more complex mixtures you have, that is a fairly valid statement to make, I guess. Right. So that concludes the Miller-Urey type studies. But there are people who do work on this. There's still doing science there in this field. So if you are a fan of something exploding, you can go there. These setups have a really high tendency to blow up. So if you want to see something explode in chemistry labs, this is where you will go. Okay. Now we will go to the characteristics of extant life and their studies in a prebiotic context. And the first characteristic that you will study is metabolism. Right? Pulki eats. He requires energy to carry out any kind of function that he wants to do. Okay, and this here is represented by this cycle, the citric acid cycle. It's called the TCA cycle or the Krebs cycle. I'm just taking a representative example here. It need not be the case for all life on Earth, but this is just a representation. Okay. This cycle is responsible for producing energy inside our body. Okay. If I am wrong, biologists, just tell me if I am wrong. Okay. Because even I don't know this stuff that much. Anyway, so this cycle is accomplished using a lot of enzymes. Okay. And if any one of you could explain what enzymes actually are in a very layman's term, so that even a class 10 guy would understand. Girl will understand. That would be very grateful. What are enzymes? Biocatalysts. Nice. So they just catalyze reactions, right? And they are very complex, extremely complex. They are usually proteins and very complex. Some of them are riboses, but we will not talk about those. Proteins. Think of them as machines that are designed to do just one specific task. And that machine is very, very, it has a very high efficiency and it will extremely advanced that you cannot even comprehend the scale on this machine work. Okay. So this cycle requires enzymes, okay, to generate energy. And most of the work that is done in the metabolism school of studies is based on some kind of cycle that will provide us energy. Okay. The goal of this school of study is to see what kind of cycles would provide energy in a prebiotic scenario. By a prebiotic scenario, I mean by using very simple molecules which are present all across the universe. Okay. And I have a representative study of that, but keep in mind this cycle. Okay. Don't remember the names, just keep in mind that this cycle exists. And you require a lot of enzymes to do it. Okay. So in this paper that I've given, that I've mentioned below, the authors have proposed a reverse cycle. So the last cycle that we saw, the one that is actually present on life, in life, the Krebs cycle, it proceeds in an oxidative manner. This here is a reductive cycle. What does that mean? It moves in the opposite direction. Okay. And the authors of this study did something very phenomenal. They managed to reproduce this cycle with the help of just complex metal ions, heavy metal ions, not complex metal ions. Heavy metal ions, which are fairly abundant on Earth, and a few other molecules which are again very abundant on Earth. And by other molecules, I mean mostly carbon. Okay. So here, if I'll show you, this ATP here represents, whenever you see a dotted arrow, it means that, okay, this dotted arrow and ATP means that you require ATP to do this. And what is ATP? It's just a molecule. Yes, you don't need to know the full form. It's just a molecule that is present in extant biology, as in present in life everywhere, and you require it to go from here to here. Okay. But the authors of this study found out that you can go very simple, right? Carbon dioxide is very common and it's very abundant across the universe. So has a lot of sources. Okay. And you can see what metal ions they took even below. Okay. So to summarize, metabolism school of studies with very simple concepts. Okay. This is what the metabolism school of studies does. Now move on to the next characteristic. All organisms replicate at one level or an organism there. Right? So he recreates. Hopefully he will. I hope not soon, but anyways. And this is the one that I find personally very interesting and it's one of my favorite things of life. Anyway, replication is just a fancy word for information transfer, okay? Information transfer as in what? So, what is the molecule that are shown in the column? DNA, here DNA. Right, that is DNA. Right? DNA is it's the coded information inside our body. Okay. The information to do everything. Has a lot of junk, that is fine. But also characteristics that could be true, all of them are included in this double sentence. But it's just coded information. It's not the actual information, right? DNA, DNA creates DNA by merging with another of its own kind, not of its own kind. That's not the main point. When we talk about information, DNA creates RNA. RNA contains the information to create proteins. Proteins are the machinery. Protein is the information that you need. Okay? And we need to transfer this information across species. Not across species, across organisms through a time scale. Okay? So the progeny of someone will have information of its ancestors. Right? That's how information is transferred across timelines. So we can see that we have evolved certain kind of coded information. We have evolved storage of information. It's easy to replicate, it's easy to say that, okay, DNA is our information, but it's not. It's just a medium of storage for that information. The actual information is proteins. So we can fairly simply say that, okay, this is the information that we need to transfer across timelines, but we have kept it coded like this. Why? What is the need to code it? Pulkit, you had something? Compactification, fine. So good answer. It's a good answer. Okay. The answer is that proteins are hard to synthesize first of all. They are very long and they are not, and there are 20 different amino acids that like life uses. And it's very difficult to find all of them at the same time. Right? The other thing is information just as it is can be corrupted easily. That is something like all of you know. Right? Just don't leave your information out in the open. Someone will definitely corrupt it. So that's why we have found out ways to code it in form of RNA and DNA. But then, why this extra step of RNA? Why not just directly from DNA to proteins? That is also another question that you can ask. And for comparison, I have shown you RNA. This is RNA. There's like two differences in the structure of DNA and RNA. That is, first of all, you can see that it has an extra alcohol group at the third position of the sugar ring. Is this too much chemistry? Do I need to point it out? Okay. So you can see that here there is an alcohol, right? This alcohol group is absent in DNA. And the other difference is that RNA always exists in single strands. DNA has two strands. Okay. So this here is a polymer, and DNA would have some. RNA generally does not do that. There are different types of RNA, we are not going into that right now. Just as itself, RNA has a single strand and it has a free OH group. And this is very important. Why? That is because RNA is not a very stable molecule. And that is due to the free OH group that we have over here. That makes it very unstable. Okay? So, this is a molecule that is capable of storing information, but it's unstable. And we have this molecule in life. How would you take a better way to store it? We evolve, right? Evolution works. Evolution works on what is already present and improves it. Right? And that's why it leads to one of the major theories in this school of study, and that is the RNA world. Being unstable is harmful because why do we require DNA? Why can't we just use RNA? Because RNA tolerant to different environments. Right? You heat it, this RNA will decompose. Right? And people who are working in labs, they can probably verify this. Yes. RNA decomposes very easily. Heated does not. Okay. So this gives rise to the RNA world hypothesis. The keystone of all the study that is done in this field of study, as in the replication part of the chemical organism. Okay. What this hypothesis tells us is that before we had DNA as the main form of storage of information, there was RNA. There was no DNA. And DNA came as a product of evolution from RNA. Is this making sense? I gave you the reason in the last slide. Does this make sense? I can't see nodding faces in the back. Does this make sense? Okay. Not you, the back. But yeah. So the RNA world hypothesis says that okay, in the beginning of whenever we started coding information, we had RNA, and from there we evolved into DNA. Okay, and some of the main proponents of this field consider viruses as a remnant of that time. Then we used RNA to code information. But anyways, again, the same case with glucose. RNA is not the beginning of time. Okay. Here you can see that RNA and DNA can be broken down into three simple components. And they are mentioned here. The recognition unit, the trifunctional connector, and the ionic linker. All of these are important. All of them play certain role in the formation of the entire chain, entire polymer of information. Which means that all three of them perform a certain function which is essential. And all of the studies that people do in this field play around with all the three of these parts. So you can substitute any of these parts with something else and you see whether it's a viable option for coding information. Okay. And then you try to guess how life would have begun coding information. Okay. That concludes this part, and this is the part that I'm most interested in. Right. This was the replication part. And the next part, which is also a major field of study, but I do not follow it too much, and it's really hard to put all of it in 1 hour. So I'll quickly gloss over it. It's compartmentalized. Okay. And this is the last of could be. So I'm really sorry I had to use you, but it's my last talk, so I figured it would be appropriate. When does could be used on it? Extremely harsh sunlight, right? So Kulkit is using the umbrella as a mean of protection against harsh elements of the environment, right? All life has something similar. We all have cell walls and cell membranes, right? These are very important because they protect. That is one of its functions. The other function is that it makes sure that whatever is required will be inside the cell. Whatever is not required will not get into the cell. Okay, this is important, right? And the other thing that it does is that it makes sure that all the things that are present inside the cell have an optimum concentration. Does this make sense? You need an optimum concentration to perform certain reactions, right? And that is what the cell wall or the cell membrane ensures. Research in this field is mostly concerned with how cell walls must have evolved, as in lipids. What are lipids? I'll put a few examples over there. These are the molecules that they've taken. Decanoic acid, for example, or octanoic acid, or oleic acid, any of those fatty acids. So, what people who work in this area work on is finding out about how these things evolve with time. Okay. I have given an example of a paper in the corner. This paper deals with how by simply dehydrating and rehydrating a simple mixture of lipids, they form vesicles. And what are vesicles? Cells. Lipids, those molecules. Those molecules form cells which selectively incorporated. Isn't that amazing? I think that is amazing. It's selectively taking in RNA molecules and leaving out the. That is very important. Right? Most of the research in this field of study does something similar. They try to see what kind of membranes would have existed and how they would have interacted with different kinds of environments. Okay. This is all about the compartmentalization field of study, the chemical origin of life studies. And these are the end of this part of this stuff, as in the introduction. There's one more field that I have not spoken about, and that is the systematics. And to be very fair, it's a very huge topic that I cannot explain, and I do not understand it completely myself. It deals with entropies. It deals with how systems evolve at large time scales, how they interact with each other, and it involves a lot of coding and simulation. I'm not good at any of those, so I didn't include it, but it's there. Yeah. If you want, you can ask me about it, and I'll point you in the right direction. But don't expect too much from me regarding that. Anyways, these are the types of molecules that we work with in the column. Right? These are ammonia, water, carbon dioxide, carbon monoxide. Very simple molecules. And you might have a question. These molecules are something like it evolved from something like this. Right? It's a very question, simple question. And the answer is that given something like this also happens, very simple to say that over the time scales of complex toolkit must have occurred. Right? You can see that just by taking two very simple molecules, they're not very far away from each other, but then react with each other to form very complex. Okay, and this is experimentally verified. You just need a little basic medium in water, that's it. You get to sugars, and sugars are very important, right? As carbohydrates, and they are also present in RNA and DNA. So these are very important molecules. If you start with simple molecules, you end up at very complex. In a very short span of time. Think about what evolution could do for millions of years. That's why we have to be worried. Okay. That explains all the fields of studies that we have in the origin of life. Now I'll be talking a little bit about my own work in this field, and it will be slightly more chemistry oriented. Okay. 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 So in the replication part, I mentioned that it's not the case that RNA is the starting of life. We can compartmentalize RNA and DNA into three parts. Okay. Those three parts are the inorganic linker, the bifunctional connector, and the replication unit. Okay. In real life, as in when we consider DNA, the inorganic linker is something called a phosphate group. Okay. Phosphate groups, very easy to understand. Bifunctional connectors, sugars. That's it. Doesn't matter which sugar. We are talking about all sugars for now. Sugars, bifunctional connectors. And recognition unit are something called linker. Sometimes we have more, but for the majority of them we have four recognition units. A, T, G, and C. You must have heard of them. Even if you are not in biology, you must have heard of them. Is there anyone who has not heard of them? Well, faster can do yes. First, so you have four recognition units for DNA, and we have four for RNA. Three of them overlap, one doesn't. Okay. There is just extra information. But anyways, you can see here that you can classify this RNA as having three components. Right? This is what we spoke about. We can play around with all of those. That is very important. Here I have shown you extent RNA. This here is the phosphate group, this green one. The red is the sugar, which is the trifunctional connector. And can be either of four, ATGC or AUGC, doesn't matter. Okay. What if we play around with this? What if I replace this phosphate group with something else? So, some people decided to exchange this phosphate group for a glycosylate group. What is a glycosylate group? It's just a molecule, not phosphate. That's it. Okay, it might be a little hard to see, but green is. There's something else here. There's not phosphate groups here. Okay. And to their amazement, they found that okay, this also forms and it has coded information within it. Because the information is stored in the nucleobases, right? When you have a diversity, you have information. If you have just a single letter repeated, you cannot have coded information. We need at least 0 and 1 to give out information, right? We have 4 of them. That is where this information comes in. So we can really play around with all of these 3 things. And to everyone's surprise, and this here is an example of DNA. DNA is nothing but DNA molecule in which the phosphate group and by a protein. Okay, so it's called protein nucleic acid. In exchange, the protein nucleic acid. Okay, and these DNAs are found in life. These DNAs are found in cyanobacteria. This is very important because it tells you that there is something other than DNA. Right? Where did this come from? If majority of life does not use this, where did this come from? That is very important. And it also points us towards the direction that okay, we can play around with this DNA. No, they have DNA. So your body will not recognize this. This molecule cannot be recognized by your body, but it's still information. Right? Because the information comes from this blue things, right? Not the red and green. You can play around with all of you can make the red and green anything that you want. It's still information. So this all leads us to something called the polymer fusion model. How I'll explain it now. So there are various ways to explain how RNA. There are multiple models to tell us how this polymer was formed from simple models. Assume that okay, one by one molecules came and got attached to a chain. That's how you usually make a polymer. Take a molecule, you add another to it, then you add. That's how most of polymer reaction works. But the polymer fusion model that also exists. It was put forward by. What this model tells us is that now this is something little complex. So if you do not understand, please ask me. And the nucleobases, they can form complex molecular assembly from there. They can stack on top of each other. Okay. What does that mean? They can form polymers on their own. They can stack on top of each other. This happens in DNA. Nucleobases stack on top of each other. Okay. Without requiring the slightly out of the scope of this project. There are advantages to this. That this is a conventional DNA or RNA. Right? Two strands, DNA, right? But some nuclear bases can form hexads. Okay. This is slightly out of context, but hexads are something like this. Each of this is a recognition hexads. 6 nuclear bases in a plane will be stacked over one another of those 6 nuclear bases. So okay, hexads exist, and for that you require non-canonical nuclear bases. That has been shown. They have a very unique helix and helix assembly are can try and see whether they join together. Okay. This model has not been demonstrated yet, quantitatively. Okay. Has been shown that this model could actually work. There's no evidence of it. This is okay. So I hope you all understood the polymer fusion model. We have not yet constructed to demonstrate this model. We have to clear this DNA and RNA. Right? And even if you clear on the DNA and RNA, all the 3 parts of it, it has not been combined with it. Not being shown. Right. So any more questions from the polymer fusion model? Aim of this is to demonstrate polymer fusion. Uh, Slightly complex, but you can ask if you do not understand. The wordings, I tell you, triazole linked polymers. Right. First, we come to triazole linked polymers. Okay. Triazole is this group. Okay, this green one. So my aim was to construct instead of phosphate in a backbone of DNA or RNA. I want to have a phosphate group. I have this triazole group. Okay, this was my goal. This is my end molecule. This is what I wanted to make. Okay, and these can be used as putative surrogate polymers on RNA. Yes. I said there are advantages to it. The advantage is that you do not need to include cellular selection on an evolution. Anyway, so this was my target molecule. I'm going to make this using this. Okay, this is what I made, and I wanted to go from here to here. Okay, and the reason for choosing triazole as a Pratima is because it's fairly Why do you ask? Why is it easy to concept triazole? These two will react in terms of copper plus 1, to give something like this, which is a triazole, right? This last thing, click chemistry. This thing on the chemistry every this reaction occurs in water. First big thing occurs in water. Second big thing, it's extremely tolerant to anything that you put in it. You just require copper 1 and these two ions. That's it. You have a water or any medium that you want. Yes, it works. That's it. The other thing is it's quantitative, as in you almost always get to 100% yield. This reaction almost always 100%. Other it is not technically so here. It will work in non-polar solvent as well. Any solvent that you want. You just require copper 1. And the biggest problem is copper 1. Even if you just take these two molecules, heat it, you will still get triangle. That is also one good thing about this. Right. I removed that slide, but yeah. Right. This was my end product. This is what I wanted to make. These things are biocompatible. So enzymes will act on that fragment as well. That is huge. Why is it not huge? Because this is something that we introduced artificially, and biology still recognize it. At least we are schooling biology to recognize it. And enzymes generally have backward. Enzymes are specific machines, they cannot move forward in time. But if you have something that worked before, enzymes will work on that as well. That works even in experiments. So why not? Okay. The aim was simple, to construct something like this. Okay. And to attempt polymerization of same monomer under copper catalyzed conditions using chemistry. Yeah, something that we catalyze. And these two are goals that I could not complete, but these two are also goals, and I will talk about them. The last one was to demonstrate polymer fusion. Then you go to the result. Keep in mind that this molecule here has assigned an alkyne both on itself. So, what does that tell us? This molecule can self-polymerize. Can create polymers on its own. You just read it, that's it. Third, okay. And even more important is the fact that three components to make DNA or RNA. And another nucleic and forget the recognition medium. If you put one molecule to two molecules, right? Very simple. So I made this molecule, and then I studied whether this would make polymers. Okay, because I did not have time to go to that molecule, see whether this polymer exists. If this polymer exists, it's still as good as the result as that. Right. So this is what I was trying to do. Slightly complex, but you can see that the molecule that is. And forget this part. This is just copper one. Okay. This here is copper sulfate, and this is a molecule that will convert the copper. That's the role of this molecule. Worry about that. This is polymers, something like this. And just by getting it, will I be getting polymers as well? So this is that part. Just by getting, would I get polymers? Okay. This part is fairly well established. Clean chemistry, Nobel Prize, everything. Okay. Right. So I did this reaction. I took this molecule. That is one of the big things about studying. That is one thing that I think about. Anyway, and this is the result. Okay. There's no way to quantify this molecule, this polymer. Why do I say that? Because this polymer is was not soluble in any solvent. So if you read spectroscopy, if you do not understand mass spectroscopy, okay, mass spectroscopy does something. If you give it a sample, it will tell you all the chemical species that are present in the sample. That's it. How does it do that? It tells the molecular mass of each of the species in the sample. Is that fairly simple enough to understand? If you have A and B, A has a molecular mass of 1 and B has put it into a mass spectrometer, it will show you a peak at 1 and at 2. It will not show a peak at 3. Okay, that's what a mass spectrometer does. Tells you exactly what chemical species are there. Okay, so whatever came out of this reaction, I ran it to a mass spectrometer. And this is the most beautiful result that
हमें
तुम नहीं करो सॉरी this is very important and to the study. Why? This molecule has a mass of 253. Okay, so this molecule has a mass of 253 atomic units. Okay, basic chemistry. What is molecular mass? This is the mass of all the atoms that are present. Right. And this is what I got. Multiple of 250. What will you get? But you do not get a peak at 500. Minus 503 is 506 is 23, which is the mass of sodium. Fairly common in mass spectroscopy. Okay. Here's what made this so interesting. You take this peak, you subtract the value of this having 11 of this in very simple conditions. Okay, so this molecule has a propensity to self-polymerize, and I wanted to see copper here. I wanted to see if this, that was the next point. And there's a definite preserver. The difference is 253, which is the mass of this one. Right? See, subtract 500 million from 782. Then you can also go one step further. 782 from 1066. And this thing happened even without using copper, which is interesting. I just heated this. Um, since it's not a catalyzed reaction, you will see that most of polar findings detectable. Right? This thing happens even without. Um, but it shows that you have molecules that are so, and by that you have already constructed like the required three, you already made two, just by the one molecule. That is simple. Right. This is the conclusion of the study. I ended here, but you can see that this in actual life, this thing does not exist. Right? You give you something like this. This is starting to look like the sugar molecule that you have in DNA or RNA. Actual aim of the study was to perform all of these reactions and to see whether polymers go outside because you are masses. Yes. Polymers. No, that's the end goal, but 2 months is not enough to do that. So, DNAके context में, when you look at this polymer, you see that it has a sugar group and you have a, you require 3 parts of the construct DNA. Two of these parts are already been constructed. Taking a simple molecule, they are taking 2 of the 3 parts. 2/3 of your job is done. You just need to add the third. And third part is like, well documented. You just need to heat this kind of molecules with the replication unit. It has a propensity to form bonds. Right? You see where I'm going? If you take a replication unit that is known for forming stacks, okay, as in it can form polymers on its own. And you take this molecule and a polymer of that molecule, something like this. Molecules have a tendency to form bonds. Technically, they want the polymer fusion bond. You can do that. That is huge. That is solving a very big problem of. Okay. Right. So yeah, one of the main end goals was to demonstrate polymer fusion. No time, so I could not do that. These are also additional goals that I. But yeah, that was the gist of the study, and this is the even by just simple reading. Okay. Like tribal links are very simple. Okay. So zero linking message is one. Okay. Right. This brings certain someone, Sudharan. He is instrumental in my career since last year. He's he's a professor there. He's in biology department, and I made and I converted part of the lab into a chemistry. I had one, she had one. Do the work her name have an interest in something like this? She is one of the few people who work on this part of studies, chemical life in astrobiology. She is very receptive. She will answer your questions and whatever you ask. Doesn't matter if you are asking her any generic question about your life, she will still answer. Okay. And it will be a very good answer. I can guarantee you that. Anyway, all of this is due to her. I was a trainer. Yeah, questions you want to discuss? Yeah.
So few words of TCS I think.
Whether you can create a certain metabolic cycle, something that would give up energy or at least take it away from other sources and give us a cycle similar to that was designed. Results are very awesome. They wanted to see if we can achieve the certain cycle with